Hello everyone. Good afternoon. It's a great pleasure to introduce speaker Stefano Fusi. Stefano is a guest at the IIT with Stefano Panzeri, who's not here, who is in... Where's Stefano? Stefano is calling me. No. No, it's my daughter calling me from China. I'll speak to him. Is Stefano listening?
Yeah, I mean, that's why we want...
It's already arranged? So okay, perfect. Okay. So it's a great pleasure to introduce Stefano, who is a person I've known for a long time. And let me say, first of all, a few things about him, and then a bit of an anecdote. Stefano studied with Daniel Amit. I think he did his PhD in Jerusalem. That's correct. And then he spent some time at Brandeis working with Larry Abbott, great computational neuroscientist, who then went to Columbia, and the first thing he did, I think, was to hire Stefano to go and work there. And Stefano now is an associate professor with tenure at Columbia University. I first met Stefano many years ago when we were starting to build CIMeC. And part of the dream of what we want to do at CIMeC was for it to be truly a cognitive neuroscience center. And cognitive neuroscience without computational neuroscience is limp. And so I asked Daniel Amit, who I didn't know at the time Stefano. Daniel Amit says, well, you should speak with Stefano Fusi. And Stefano came, and he has talks here. We tried desperately to convince him to come, but we couldn't compete with Columbia, so he went to Columbia. But that doesn't mean we cannot get him to come. So we're still working to see if we can convince him to come back to Italy, and perhaps some joint appointment between IIT and CIMeC. In any case, Stefano is doing some beautiful work in computational neuroscience. The data that he's been using lately is single-cell recording he obtained with Earl Miller at MIT, and so, welcome, Stefano.
Okay, thanks a lot, Alfonso. I mean, I'm really glad to be here in this beautiful palace. And yeah, I was here about 10 years ago visiting Alfonso, and it's great to see how his creature has grown up, and now it's really a fantastic place here. Okay, so what I'm going to talk about today is these high-dimensional neural representations. So, probably even the title is unclear, but don't worry, because I will go over every concept and I will try to explain everything from the very beginning. What I have in mind is essentially to explain the messiness of the brain. Okay, so this is a beautiful depiction. Can we lower the lights a bit? Just for this slide. Okay, it doesn't matter. I mean, you probably get the idea anyway. So when you stick an electrode in many brain areas, and this is particularly true in prefrontal cortex, you really see so many diverse responses of the neurons. It seems that they respond to all possible combinations of the task-relevant variables. And this is beautifully depicted here in this painting by Umberto Boccioni, which probably most of you already know. And it's not by chance that the title is States of Mind. This is a part of a triptych that you can see at the MoMA in New York. Now, I don't know how much you can see it, but this is essentially depicting a farewell. So, there is a man and a woman, they are embracing. You can see here, you can see essentially this man and woman from many different angles, representing somehow emotions and memories at different times. And then you see a locomotive coming, the same locomotive seen from the side, and everything is completely mixed up. And I believe that this is really what you observe in the brain. So, many electrophysiologists are trying to find neurons that are highly specialized, what I'll try to show you is that these kind of representations where the neurons are highly specialized at encoding only one variables are actually bad representation. And there are a lot of things you cannot do with these representations. Whereas these very mixed representations are those that would actually enable you to have such an incredible flexibility in performing so many different tasks. So that's why it's so important in prefrontal cortex. Okay, so let me try to be more formal and to exactly define what I mean by mixed selectivity. Then I'll try to show you that this mixed selectivity is related to the geometry of the neural representations. And that's why I want to talk about dimensionality. And then I'll show you why this is important. And then finally, I'll show you some data that actually prove that this kind of high dimensional representations, where you have a lot of nonlinear mixed selectivity, are actually fundamental for performing some task. And the neurons that are highly specialized very often are there just by chance. They're really a minority of the neurons, and for good reasons. So in the very last part of the seminar, I'll try to present some recent ideas on how to estimate the dimensionality of these neural representations using fMRI. Okay, so let me start with a very simple example. So I want to start from single neuron properties. So let's consider the following problem. You have a task where you have to classify different stimuli, and the stimuli have two components. So you can imagine a stimulus that has a visual and an auditory component. And let's say A and B are the two parameters characterizing these two components. So you can imagine that A is the luminance of the stimulus, of the visual component, and B is the sound intensity. Okay, so now let's say you go as an electrophysiologist to the brain and you record the activity. And this would be one particular neuron where you see the firing rate. This is just a contour plot. You see the firing rate as a function of the two parameters, A along the horizontal axis and B along the vertical axis. And you see that the firing rate of this neuron increases only along this direction. So basically, this is a neuron that is highly specialized. Okay? So it's a neuron that knows everything about the visual component, and its activity is not modulated at all by the parameter that characterizes the other component, the sound intensity. Analogously, you can define a neuron that is highly specialized at encoding the other parameters. So it would be just rotated by 90 degrees. Okay, so this is what I would define as pure selectivity neurons. They care only about one variable. Now, you can imagine a very simple situation where the response of the neuron, it can actually be described as a linear function of the two parameters, A and B. So that would be a contour plot that is very similar to these two. It would be just rotated. And this is what I would call a linear mixed selectivity neuron. Okay, now whatever is not described by these kind of models of very simple neuronal responses is what I'm going to define as nonlinear mixed selectivity. And here there are a couple of examples. So the first one is a neuron where the firing rate essentially increases if you move along this direction. And you see that this is clearly a nonlinear function, and this would be a different nonlinear function. Of course, real neurons would be much more noisy, but here, I'm just trying to give you the definitions. So I'm considering noiseless cases. Okay, so in these cases, the information about the two parameters is distributed across neurons. This is typical of these representations. If I record from the first neuron and I get 5 hertz, I cannot say much about A and B because I get 5 hertz along this entire line. And I need multiple neurons in order to decode these variables individually. So these are not easily interpretable response functions. And these kind of neurons have often been ignored in the analysis simply because they cannot be described in simple terms. So what I'll try to show you is that these are the important guys in most of the tasks that you can imagine. And in order to explain that, this is something that is well known in the machine learning community, but I'll try to summarize the main concepts here. And in order to prove this statement that I've just made, I need to start with some considerations about the geometry of these neural representations. Okay, so let me start from this kind of representations that very often electrophysiologists are looking for. Pure selectivity neurons, highly specialized neurons that know only about one variable at a time. Okay, so let's pick 3 of these neurons. They will be F1, F2, and F3 will be the firing rates, and there are some linear function of the different parameters, okay? And I'm going to use a lot this representation, so please stop me if it's unclear. So I'm going to consider the firing rate space. And along the coordinate axis, I'm going to have the firing rate of all the neurons I'm considering. In this case, I have 3 neurons, so I'm going to have F1 along this x-axis, F2 along the y-axis, and F3 along the z-axis. If I have n neurons, I'm going to have an n-dimensional space. Okay, so now let's say I run my experiment. I show one stimulus, and I record the activity of these 3 neurons. Then I get a triplet of firing rates that essentially can be represented as a point in this n-dimensional space. In this case, it's just a 3D space. And I'm going to represent the point as a sphere just to make it more visible. Okay? So let's say A1 and B1 are the 2 parameters that characterize stimulus A. Then I'm going to get F1, F2, and F3, which of course will be function of the stimulus, the particular stimulus that I'm considering. Okay, I repeat the experiment. And if I'm in an interesting area, brain area, then I will get a different response. And so, for instance, a second point here. And then a third point corresponding to a third stimulus, and then a fourth point. Okay, it doesn't matter how many stimuli you consider, there is a property that all these sets of points share, which is the following, that these 4 points will be contained by a plane. And now I can introduce the dimensionality. So what I mean by dimensionality here is the minimal number of coordinate axes that allow me to specify the position of all the points that I'm considering. So in this particular case, the points are on a plane, so all I need is 2 properly aligned coordinate axes. So the dimensionality of this representation is only 2D. I'm saying only because this is lower than what it could be here, which would be 3D, and I'm going to discuss that in a second. Okay, before going to the high dimensional representations, what happens if you replace one of these neurons, let's say the third one, with a linear mixed selectivity neuron? Basically nothing. The geometry will be exactly the same. The plane will be tilted, but you will still have a 2D representation. And that's why I put the linear mixed selectivity neurons on the same slide as the pure selectivity neurons. Okay, so why do we care about these kind of properties? The reason is that we can probably define some kind of quality, measure of quality of these neural representations. And for instance, I can take the point of view of a very simple readout, let's say a neuron that is downstream, and is reading out these 3 neurons. So now these 3 neurons represented in the firing rate space are the inputs, and I'm going to take an output that is reading out these neurons. Okay, let's say I have these 4 points, and then I want to train the readout to respond to the stimulus that corresponds to this point and not to the other 3. Okay, that's why this is yellow, the other 3 are red. Okay, that's something I can do very easily because I can just use a linear readout. And a linear readout is equivalent to drawing a plane, and more generally an hyperplane, in this firing rate space that separates the yellow point from the red points. So this is easy, I can do it. Okay, so now what happens with a more complex situation? So let's say I want to solve this other problem. I still have the same neural representations, the input is the same, but now the desired output is different. And many of you have probably recognized this configuration as a configuration that is equivalent to the exclusive OR problem, which is known to be non-linearly separable. So here, in order to solve this task, I need a more complex readout that would correspond to a curvilinear surface, something like this. Okay? I cannot do it with a linear readout. This is the main limitation of this kind of low-dimensional neural representations. Now, if I go to the beautiful world of nonlinear mixed selectivity, and now I introduce a neuron, F3, whose firing rate is a nonlinear function of the two parameters A and B, now I have that these vectors that essentially define the position of the points span a 3D object, a tetrahedron. Okay, so this is in 3D space. So now the dimensionality is 3, and this is the maximal dimensionality in this case. And the beautiful thing of this high dimensional representation is that I can do any classification I like. So in particular, the one that I couldn't do with the linear classifier. And if it's unclear, I can show you here with the movie. I guess I have to do it like this. That the two yellow points are on one side of the plane and the red points are on the other side. When you have these high dimensional representations, you can do any binary classification of the inputs. And that's why, you know, the support vector machines had such a huge success, because they use exactly this trick. They project the representation into a high dimensional space, and then they can perform any task that you want. Okay, so let me summarize this part that I believe is familiar to most of you. That's why I went relatively quickly over these concepts. So if you have that the response properties of the neurons, you have this kind of pure or linear mixed selectivity, then you have low dimensionality. And in order to perform some task, you need a complex readout. Okay? So probably, you know, a few layers downstream. If you have nonlinear mixed selectivity, then you have high dimensionality, so the geometry is different. And with a simple linear readout, you can do whatever you want. Okay, so it would be nice to record the activity in different brain areas and characterize more quantitatively the quality of these different neural representations. But that's something that is difficult. And it's probably easier to take the following approach and say, let's limit the complexity of your readout. And the choice for now is going to be necessarily arbitrary. So let's say I choose the linear readout, okay, which is simple enough. Of course, if the readout is too complex, I'm not going to learn anything about the neural representations, because you can imagine a readout that is as complex as the brain, then it can perform any task that you want on the input. So then, you know, we'll not be able to discriminate between different brain areas. So I need to limit the complexity. I choose arbitrarily the linear readout. I'll show you later that this choice is not as arbitrary as it might sound. Okay, so I choose this linear readout, and then I simply count how many of these binary classifications are implementable by the linear readout. In this case, there is clearly one, and actually there is another one where you swap red with yellow that cannot be implemented by the linear readout. Whereas here, I can implement all of them. Okay, so in the general case, I'm going to have n neurons, which would be the number of axes. And let's say p would be the number of spheres. And I'm going to consider the case in which p is smaller than n. So the number of stimuli or the number of experimental conditions you want to analyze is smaller than the number of neurons you're going to record. Okay, so then in this case, the maximal dimensionality is set by p, which is the number of conditions or points in this n-dimensional space. And then what I do is to come up with one particular coloring, and I check whether the points will be linearly separable or not. Okay, this is just a cartoon. You have to imagine this in an n-dimensional space. So, you know, let's say that in an n-dimensional space, this would be actually linearly separable, so I put this mark here. Then I go over another coloring, and let's say this is not linearly separable, and so on and so forth. I go over all possible 2 to p colorings of the points, and I just count the number of binary classifications that are implementable by a simple linear readout. If this number is large, it's a good neural representation. If it's small, then it's limited. Okay, so you can compute this NC as a function of the dimensionality, and you find that it's basically an exponential function. Okay? This is telling you two things. The first one is that you have a huge advantage at using high-dimensional neural representations. And the second one is that you can measure either NC or the dimensionality, and then there is a simple relation between the two. And actually, this is the strategy we're going to adopt to estimate the dimensionality. We will actually measure the number of binary classifications that are implementable by a simple linear readout. Okay, so going back to this very simple scheme, I have here pure and linear mixed selectivity, low dimensionality, NC would be small, whereas for high dimensionality, NC would actually grow exponentially with the dimensionality of the neural representations. Okay, so this kind of numbers, in particular NC, is something that you can measure from the data in experiments. And that's something that we did. We did it initially in the data of, using the data of R. Miller, that's already published, but more recently we did it also in many other brain areas, part of prefrontal cortex, in the amygdala, in the hippocampus, and we find very similar results. I will not have time to discuss that, but if you're interested, then you can come later and ask me. Okay, so let's start from prefrontal cortex. This is a beautiful experiment that has been performed in the laboratory of R. Miller by Melissa Warden, and the original paper came out in 2010. And they trained a monkey to perform a memory task. So there was a sequence of 2 stimuli, visual cues. First cue, then a delay, then a second cue would appear on the screen. And the monkey had to remember this sequence. And there were 2 ways of testing the memory of the monkey. The first one was with a recognition task that I'm going to define in a second. The second one was with a recall task. So they were in blocks. So actually the monkey would know always what kind of task would perform from the very beginning of the trial. Okay, the recognition task was like this. So there was the sample sequence, the one that I've just shown you, followed either by match or a non-match sequence. Okay? So a match sequence is where you have both stimuli that are identical to the sequence that you have in the sample sequence. Okay, the monkey had to hold the bar until it would recognize a matching sequence. And after a non-matching sequence, there was always a matching sequence. The recall task, we had the same sample sequence, but then 3 stimuli would appear simultaneously on the screen, and the monkey had to make saccadic movements to the 2 stimuli of the sample sequence in the same temporal order. Okay, so
just to clarify, for both experiments, I don't remember the details. For both experiments, these were not associated pairs, but rather novel presentations.
Exactly. They were novel in every trial, and actually even the stimuli changed in every session. So there was a set of 4 stimuli that would be different in every session. And then, yes, the stimuli were completely random. So there was, they couldn't rely on long-term memory to essentially perform the task. Okay, so now if you record the activity from prefrontal cortex, this is a typical neuron that you would observe, okay, with this kind of mixed selectivity. So this is a classical PSTH. Every curve is the firing rate of the neuron as a function of time. And this is the time from Q1 onset, so this is the presentation of Q1, and here you have the presentation of Q2. And here I didn't even list all possible conditions because there were too many and it would be too crowded. But what you can see is that the selectivity of the neuron is not an inherent property of the cell. It changes depending on the epoch of the trial, and it's a lot variable. And actually the neuron doesn't have strong preference for a particular stimulus or for a particular task type or for a particular combination of Q1 and Q2. Typically, it responds to a nonlinear combination of all these different variables. So you can see here where the yellow arrow is, this would be the firing rate of that neuron in response to some of the conditions. And you see that you always have this kind of complicated profile. Okay, you might wonder maybe this is just noise, but it's actually reproducible enough that you can rely on this to decode all the task-relevant variables. Okay, so let's go directly. You can find many more details in the paper, but let me go directly to the dimensionality analysis. Okay, so they recorded from 237 neurons. Not all of them, obviously, were simultaneously recorded. And this was in this 800 milliseconds time interval. So where presumably all the information about Q1, Q2, and task type would be available. Okay, so there were 4 objects for Q1, same 4 objects for Q2, but Q2 could not be the same as Q1. So actually only 3 out of 4 could be chosen for Q2, and 2 different task types. So the total number of conditions was 24, which is also the maximal dimensionality that you can have in this n-dimensional space, because you have 24 points in this 237-dimensional space. Okay, so this is summarized here. So you have 237 coordinate axes and 24 spheres. And so you have 2 to 24, which is about 17 million possible binary classifications. You can go over all of them and see whether they are implementable or not. And what I mean by implementable is that you want to make sure that you train a linear decoder, a linear classifier, on some set of trials, and then you test it on a different set of trials. So it's all cross-validated, so it's real. We're not cheating in any possible way.
Sorry, Stefano, I missed something here. So there's 4 times 3, I'm missing another factor of 2. Is that because you reverse the order? I'm trying to figure out how you got to 24.
Number 2 is what is unclear.
Yeah.
So because there are 2 tasks. So there is the recognition task and the recall task. So, this sequence can be followed either by the recognition task or by the recall task. The monkey knows that from the very beginning of the trial, because these are in blocks. These are, 150, 180 trials blocks. Okay, so these are the main results, at least for now. So here on the y-axis, I have the number of possible binary classifications. And on the x-axis, I have the number of neurons. Now, these numbers are much larger than the actual number of neurons that we had, simply because we wanted to reach an asymptote for this curve here. Because, when you're unable to perform a classification task, there could be 2 reasons. The first one is that the dimensionality is low. The second one is that the dimensionality is high, but you have too much noise. So in order to make sure that the limit is really due to some limitation in dimensionality, you want to take the limit for the noise that goes to 0, and then you hope that this happens when the number of neurons goes to infinity. Now, we could have done the entire analysis with the neurons that we had, but we wanted to reach the asymptote, so we just resampled the neurons in a way that, we were very careful that does not increase the dimensionality. Okay, so here you see 2 curves. The black one is for the actual data, and this, just as a reference, is for pure selectivity neurons. Okay, so imagine that you have neurons that are highly specialized at encoding Q1, neurons that encode Q2, and neurons that encode task type. You pull them together and you read them out. Okay, this is the best that you can do. Okay, the maximal dimensionality is 7. You have to trust me, but it can be computed very easily. And this means that the maximum number of binary classifications you can do is only 128. Okay, now these were pure selectivity neurons with the same noise as the real data. So you also need some neurons to reach the asymptote. This was the real data. You see that it goes all the way up to 24 dimensions, which is the maximum. And you see also that there is a huge advantage in terms of the number of classification tasks that you can perform if you use the actual data instead of this kind of patchwork of highly specialized neurons. Okay, so here we were wondering whether this kind of dimensionality is actually used by the monkey. Okay, it could be just an accident. It could be purely coincidental that we found this high dimensionality. Now, in order to do that, to verify that actually the monkey is using dimensionality, the ideal experiment would be to shut down all the weird guys, all the neurons with nonlinear mixed selectivity. But that's something that is extremely difficult. We've been thinking a lot about it, and we still don't know how to do it. However, we could do something here. We could analyze the errors. And the idea was to see whether there was any correlation between dimensionality and errors, which would indicate, just indicate that dimensionality could probably be important for performing the task. And actually, that's what we found. Every time the dimensionality was collapsing, the monkey was going to make a mistake. Okay. So we couldn't do the analysis of the errors for all the 2 tasks because we didn't have enough errors. But fortunately, the monkey did a sufficient number of errors in the recall task. So now we're going to restrict the analysis only to the recall task, where we have 12 different conditions, so the maximal dimensionality will be 12. And in this case, you see that the black line is for the correct trials. You go all the way up to the maximal dimensionality. And you see that for the error trials, this collapses. It goes down. Okay, now there could be a lot of trivial reasons why the dimensionality collapses. Okay, so for instance, let's say the monkey didn't see one stimulus, didn't see Q1. So in that case, of course, you would fail training a linear readout simply because you don't have the information about that stimulus. However, this was not the case, okay? And we were lucky because it didn't have to be like this. I mean, it could have been that the monkey was making these kind of trivial errors, but it was not the case because when we were trying to decode Q1 and Q2 individually in the correct and in the error trials, we got exactly the same performance. Okay, so here you see this is the decoding accuracy for Q1. The green line goes up. Here it's almost 100%, and then it goes down, but it's still significantly above chance level. And this is for Q2. And you see that the dashed line is for the error trials, the solid line is for the correct trials, and they're almost identical. So the monkey, or at least the neural activity, contains information about Q1 and Q2. But somehow this information is not in the correct format. It's in the low-dimensional format that would impair the monkey in that particular trial at choosing the right response. Okay, so we wanted to make sure that it's really the nonlinear component that is responsible for this collapse in the dimensionality. So what we did was to analyze the response of the neurons and separate the linear from the nonlinear component. So we just fitted a linear model to the response of the neuron. So the linear model will be indicated here. It's like in the ANOVA. And basically, here, this term will be the interaction term. So it's whatever is not explained by the linear model. And in this particular case, the linear model can account up to 6 dimensions, and the nonlinear component for the other remaining 6 dimensions. Now, it's important to stress that the linear component accounts for something like 85% of the variance. So many people would stop there. They would say, I have a good model. I don't really need the nonlinear component. However, as I will show you in a second, it's only when you look at the nonlinear component that you can predict the behavior of the monkey. So the important component is probably the nonlinear one. Okay, so this is essentially the linear component, the one that you would have with only linear mixed selectivity neurons or pure selectivity neurons. And again, the black line is for correct trials, the gray line is for the error trials, and you see that they're almost identical. So no difference. Okay, so the linear component is perfectly preserved. Whereas the nonlinear mixed selectivity component really accounts for the entire collapse in the dimensionality. Okay, so at this point, what I would like to conclude is that the neural representations of prefrontal cortex are high dimensional. Okay, and this is really a usable dimensionality. It's not noise because we can use it with a linear decoder and everything is cross-validated. And in order to have high dimensionality, you need a very simple recipe. You need to combine nonlinear mixed selectivity and diversity. Okay, you need both components, not just one, because you need very diverse responses that are all nonlinear mixed selectivity responses. Okay, so here it seems that the dimensionality is correlated with behavior. So you remember that I mentioned that this assumption about the linear readout was arbitrary. It was, but fortunately, it turned out that it's compatible with the readout used by the monkey, because at least when we use the linear readout, we can predict the behavior of the monkey. Okay, of course, there would be a full spectrum of different readouts that would also be compatible with the behavior of the monkey. But I can tell you that if I use a nonlinear readout, for instance, that will not be compatible with the behavior of the monkey because the information about Q1 and Q2 is available in the error trials. So the nonlinear classifier would actually always perform correct using the data that was recorded. Okay, so I want to play this game now with fMRI. And this is just an idea. We don't have any data. And that's one of the main reasons why I'm here, because I would like to interact with fMRI people, and I would like to learn about fMRI. So, this is the last part of the seminar, and try to go quickly over it. But we started a bit late, like 10 minutes late, so I can probably have another 20 minutes. Okay, now, here we have good news and bad news. Okay, so from this analysis that I showed you, we know that it's important to look at these weird guys, the neurons with very diverse responses. Now, this is a problem, because when you do fMRI, you take the average over thousands of neurons, you're probably averaging out an important component. On the other hand, you have to keep in mind that it doesn't really make much sense to do what many electrophysiologists have been doing for a long time, like counting how many neurons are selective to one particular variable. What really matters here is what happens at the population level, because the dimensionality is a global property of the geometrical representations. If you look at individual neurons, you cannot say much about what's going on in that particular brain area. So there is some hope that using fMRI, which is a way to read out a global property of a population of neurons, you can estimate this dimensionality. Okay, so I don't have time to discuss all possible approaches, so I want to focus only on one, which is probably the most controversial, but it's also the one that would probably enable us to estimate dimensionality with the same resolution that you would have with electrophysiology. Okay, so you remember these were the representations that we used in order to explain the dimensionality. Okay, so the dimensionality is a property of a set of points, and there are two ways of specifying the dimensionality or the geometry of this representation. So one is to give me all the vectors. Okay, so that's what we did in the electrophysiology experiments, because we had individual neurons. So we had the coordinates of each of these points. We had the x, the y, and the z axis. So f1, f2, and f3. Okay, this is one way. And then once you have all these vectors, you can collect them in a matrix. Okay, you can build a matrix where the columns are these vectors. So f represents the firing rate. This is the symbol for a vector. And s1, s2, sp are the different p stimuli. Okay, so now more technically, this would be an n by p matrix, where n is the number of neurons and p is the number of points, or number of conditions, or number of stimuli. And then the dimensionality would simply be the rank of this matrix. Okay, so that would be the number of, the minimal number of coordinate axes you need to specify the position of all the points. Okay, there is an alternative way of estimating the same quantity, which is the following. If instead of giving me all the vectors, you give me the relative distances or some similarity measure of the neural representations, I can also estimate the dimensionality. And the reason is the following, that I can build this matrix here, F transpose F, which is simply the matrix with all the scalar products between these neural representations. Okay, so I have S1, 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 S2, and so on and so forth up to S1, SP. Okay, and on the diagonal, I would just have all the S1, S1, S2, S2 up to SP, SP. Okay, so this is just F transpose F. It's basically the correlation matrix. And this matrix here has exactly the same rank as this matrix. So if you give me this P by P matrix, I can also tell you something about the dimensionality of these neural representations. Okay, so there is one way that, at least in principle, should enable you to estimate all the terms of this matrix. And that's something that was introduced many years ago now, in 1999, by Rafi Malach and all his colleagues. And we are now working a bit with Kalanit Grill-Spector, who is in Stanford, to try to understand what is the best protocol. But, you're probably all familiar with this idea. But we can estimate the similarity between two neural representations by using sequences of stimuli. So in particular, by estimating the suppression that you have when you present a stimulus twice, or when you have a sequence of two stimuli. So when a stimulus is preceded by another stimulus. Okay, let me summarize the idea in the following way. So let's say here you have time, you present stimulus A. Okay, and I'm starting from a very simple example. Here I have 3 neurons, and let's say the stimulus A activates neuron 1 and 2 and not neuron 3. Okay, so these are the 2 neurons that are activated. I'm going to get some ball signal that will depend on the activity of these neurons, hopefully. Okay, so let's say I present the stimulus again, then I'm going to have some suppression. This is observed in a lot of different brain areas, and it's probably due to several reasons, to input fatigue, neuronal fatigue, adaptation. There are many reasons for this. But it's something that is robustly observed in many brain areas. Of course, there are a lot of complications you're all aware of probably, and maybe if I will time, I will explain them later. But for now, just let me give you the simplest version because I want to get to the main ideas. Then we will discuss the complications. Okay, so people know that this corresponds also to a suppression of the ball signal. Okay, if I now represent this again in this firing rate space, this would be a point. And when I present again the same stimulus, I'm going to activate the same neurons. I'm going to get some suppression. Okay, what happens if I now have a different situation where SA is preceded by a stimulus that is very different from stimulus SA? So let's say it activates this neuron, but not the other 2. So the overlap here is really minimal. Depending on how you define it, it can be 0. It's an orthogonal stimulus. Okay, so now if I present stimulus A, I'm going to have exactly the same response that I would have when I present stimulus A as the first stimulus. Okay, so basically there is no suppression here. Okay, so this is telling you that if the overlap is very large, in this case it's maximal because this is identical to this, the suppression is also large. And if the overlap is small, the suppression is also small. So there is a relation between the overlap and the suppression that you have when you repeat the presentation of a stimulus. Okay, let's try to be more quantitative. And in order to do that, I need some electrophysiology again, because I want to know what single neurons do. So, there is a series of papers by Rufin Vogels, and where he essentially recorded from single units, and he studied exactly this phenomenon of repetition suppression. And typically he has a bunch of stimuli, and he looks at all possible sequences. So this would be the first stimulus, this the second stimulus, and then this would be one particular neuron. You see the firing rate response to all these combinations. Okay, so here along the diagonal, you should have the maximal suppression. Here, the suppression should be smaller, and it should depend on the similarity between the different stimuli. Okay, so they studied systematically this kind of experiments, and they came up with a few models, actually with 3 models to describe the data. I don't have time to go over all the models, so I'm going to focus on one, which is the one that I like the most. It's only the one that works perfectly with our hypothesis. But of course, then we can discuss the other models. Okay, so what very simple, and it's an entirely phenomenological model, so it reproduces the data. Okay, that's something you should keep in mind. It's something like this. So the firing rate of one particular neuron in response to the sequence AB would be the response of the neuron to SB when it's presented in isolation, multiplied by a suppression factor that depends on the response of the same neuron to the stimulus that is preceding SB. Okay, so it's 1 minus alpha, FISA. Okay, this is one way of representing it. Okay, so this is a relatively good model. There are other models where they introduce also the input fatigue, and there are hybrid models as they define them, where you have both input fatigue and neuronal fatigue, and they claim that these are the best models. I have to say that the difference in terms of variance explained is really small. But anyway, you should see the original paper. It's a very long paper. Okay, let me focus on this one because this allows me to make very simple considerations. Okay, it's not too surprising you have something like this. Okay, let's say you use a very simple model for the neuron. So let's say the response to SA is just some nonlinear function phi of the total synaptic current. And let's say this nonlinear function is just an exponential. This is now very often used because it gives you a log normal distribution of the firing rates, which is observed in many brain areas. If you have something like this, like that, and then you present stimulus A, and then you present a second stimulus, you're going to have some adaptation. So this is the neuronal fatigue, okay? And that will be proportional to the firing rate of the neuron. And very often people attribute this to the influx of calcium that introduces this negative current. If you do that, then you're going to have ISB in response to B, when B is presented after SA, minus this term, the adaptation term. And then you get something like this. So now if you expand the exponential, you get this expression. Okay, so it's not too surprising that this is a reasonable model. Okay, so now let's say I use this model, okay? And I go back to this repetition suppression paradigm. So what I want to estimate is this matrix. Now I do this repetition suppression, okay? And I assume that I can describe the response to SB using the formula that Vogels and colleagues have used to describe their data. Okay, and now I'm going to make another big assumption, which is that the bold signal is just the sum over all the neurons contained in a voxel, the firing rates of the neurons contained in a voxel. Of course, there could be nonlinearities, it's something we are aware we are studying. Okay, so then the bold in response to this sequence, so when you have SB, is going to be the sum of all this FISB term, which comes from this 1 multiplied by this, minus alpha, the sum over all the neurons in the voxel of FISA, FISB. Okay, this is just the response to B when B is presented first or in isolation. Okay, so it's something I can also measure. So then bold AB minus bold B, which is just the suppression actually, is going to be equal to this scalar product. Okay, the vector F multiplied in response to SA multiplied by the vector F in response to SB. Okay, so now you recognize this. This is exactly the term I have in the matrix. Okay? So in principle, and of course there are a lot of issues here, in principle, I can estimate the dimensionality using a simple repetition suppression protocol with the same resolution that you would have with electrophysiology. Okay, let me show you one very simple example and then we are done. Okay, so the kind of things that you can do and the kind of scientific questions you can ask in this kind of framework. Okay, so this is just an example. Don't take it too seriously. And I don't want to make any statement about this scientific problem of multisensory integration. It's just an illustrative example. That's why I'm showing a very old paper, so that is entirely based on anatomy. So here, they were focusing on the visual and the auditory component. So here you have the primary visual area, here the primary auditory area. This is in humans. And then you have where most of the projections go. So from here you would go here and here, and then every step you move one step farther. So then from here this becomes black, and this will be the projection to the next step, and so on and so forth. Okay, analogously for the auditory component. Okay, so what do we expect in this kind of situation? So this is a nice situation because we have two components that eventually have to be mixed up, have to be combined together in order to perform a task that depends on both of them. Okay, so now you can imagine that in this brain area, the responses are dominated by the visual component. In this brain area are dominated by the auditory component. And maybe in this area where the projections overlap, you're going to have both representations. Okay, you're going to have representations of both the auditory and the visual component. Okay, so let's say I have now P different visual stimuli and P sensory components, P sensory stimuli, auditory, this should be auditory. Okay, so in total I have P square combinations of all these auditory and visual components. So in terms of dimensionality, I know that here with P visual stimuli, let's assume that they are not modulated by the auditory component, the maximal dimensionality will be P. which is the number of points. Analogously here, I'm going to have that the maximal dimensionality is again P. Okay, now here, for this brane area or any other brane area where the two variables are mixed, I can have at least two different scenarios. Okay, one is where both components are present. I have the information about them, but they are in this low-dimensional format. I have these highly specialized neurons, or I have just linear mixed selectivity. Okay? And in this case, the maximal dimensionality would be 2p minus 1. It's very easy to compute it, but I don't have time to go over the details. Okay, there could be a different scenario where you actually have all these interaction terms, and you have enough diversity that the nonlinear mixed selectivity component is strong enough that you get the maximal dimensionality. And in this case, the maximal dimensionality would be p squared, the total number of combinations of auditory and visual component. Okay, now our prediction is that these are the important areas, not this one. So, our dream is to prepare a map of the dimensionality of the entire brain. In principle, with fMRI, you can do it. And then say, this particular brain area has the maximal dimensionality. Let's verify that is the most correlated to behavior that would enable us to predict when the subject is going to make a mistake. Okay, so, these are very different representations, and it would be very difficult to discriminate between them if you use any traditional approach, because the information about the two components is present in both cases. It's just in a different format. Okay, so let me build a very simple model to simulate this system. And let's say, I have a very simple population here representing the auditory component, then a visual component, and this is whatever is not under control in the experiment, and of course it will be present in the brain. And let's say these are the neurons within the voxel, and I'm just modeling them with a very simple nonlinear function of the weighted sum of the inputs minus some threshold plus noise. And I need to make some assumption about the weights. One way to expand the dimensionality is to use just random weights, and it actually does a pretty decent job at modeling the data of R. Miller, although if you introduce some learning, you do slightly better. So let's assume that these are random weights. It's just a very simple toy model to make some considerations. Okay, and I'm also assuming that these are random and uncorrelated patterns. When you have different auditory components, you're going to have different random patterns. Okay, the ball signal, again, is going to be just a linear sum of the firing rates, and then plus some noise. And I'm going to assume that I have this kind of adaptation, okay, so the simple one that I described previously, which would be compatible with the Vogel's model when alpha is small enough. Okay, so let's say now I start my experiment with only the auditory component. Okay, I activate this population, but not this one. And I'm assuming here that I have 4 different stimuli, 4 different tones, for instance. Okay? So in that case, if I remove the offset, I know that the maximal dimensionality is 3, and actually that's what I get in these simulations, both for the dimensionality estimated from the firing rates and from the ball signal. Here, what I'm showing is the percentage of variance explained by essentially all the principal components. I do PCA here, and I compute the percentage of variance explained by each component. So, or in other words, if you want, this is the value of the eigenvalue. This is the number of the eigenvalue, and this is would be the eigenvalue. Okay, so the first 3 would be very strong, and they would correspond to your ability of encoding the auditory component, and then it would go down. Okay, so this would be for the firing rates. For the bold, I'm assuming I have a lot of noise. We took it from the literature, so the curve is a bit smoother than in the case of the firing rates. But still, you can tell that these 3 guys are above these other 2, and the same for the red line. Okay, so now here we know what's the maximal dimensionality. If we know that this is an auditory area, so where stimuli are encoded, and I can do MVPA or whatever you like to assess whether these stimuli are encoded or not, then I know exactly the dimensionality. So I can use these to calibrate essentially my eigenvalues. And I can say that this is essentially the noise level that corresponds to the fourth eigenvalue. And I'm going to use this as a noise level also when I'm going to introduce the second component. Okay, so now this is one first scenario. So this could be in some brain area, not in some others. And so here I have auditory and visual that are mixed linearly. Okay, so here, in the model, this phi, the function, it will be just linear. So just linear mixed selectivity. And in this case, the maximal dimensionality is 6. And you see that these are the high eigenvalues, and then it would go down. And you see that all these eigenvalues are below the noise level that we determine by calibrating our system. Now, if I introduce the nonlinearity, all these eigenvalues would go above this line, telling you that you now reach the maximal dimensionality. Okay, so this will be the candidate area for mixing these things nonlinearly and actually performing the task. Okay, this is just to show you that you can play with a lot of variants of the model. You can introduce the input fatigue. You can introduce many different models that Vogel's used. You can introduce different types of noise. You can introduce nonlinearities, and you get similar results because dimensionality is a relatively robust quantity. It doesn't depend too much on the details of the geometry of the neural representations. Okay, so this is just to tell you that there is one experiment where people already attempted to do something like that with repetition suppression, and it's by Aguirre and colleagues. And they actually had 2 dimensions, color and shape of these different stimuli. And they were focusing on essentially nonlinear mixed selectivity. They called it conjoint tuning, but it's basically the same. So they look only at one aspect of the dimensionality. But this is telling you that it can be done, that probably the signal-to-noise ratio that you get is such that you can do something. They introduced a very complicated quantity that has nothing to do with the dimensionality, but still they could reach some results, and I don't have time now to go over this. Okay, so, this would be the ideal experiment, and this is the last slide. Okay, so this is essentially what we proposed for the Brain Initiative, and the proposal has not been funded, but we're going to do it at some point anyway. Okay, so the idea is the following. You can come up with any situation where you have that your stimuli are characterized by multiple variables. You need at least 2. Okay, so let's say 2 features of a stimulus. It could be some faces on some landscapes. So, one would be faces and the other one would be landscapes. So a stimulus would be a combination of these 2 things, a face on a particular landscape. And then you have a sequence of 3 stimuli, S1, S2, and S3. I'm going to tell you in a second why S3. And for each combination of the features, so let's say, for instance, here you have a particular sequence, A1, A2, A1, A2. You want a specific response from the subject. And the response should be chosen in such a way that you maximize the dimensionality that is required in order to perform the task. And that's something that we can control easily. For instance, in all these cases, if you just have 2 values for each feature, so this would be binary numbers, you can have that the response is the parity function. So you just take the sum of all the ones. If it's odd, you want 1 response. Let's say move a lever to the right. And if it's even, then you want to move the lever to the left. And that's guaranteed to maximize the dimensionality. Okay, the reason why we want to have the 3rd stimulus, because 2 stimuli would be sufficient to do repetition suppression, is that we know from electrophysiology that as soon as you present stimulus S2, the subject can generate the response in 20 to 50 milliseconds. It's really short. And once you have the response, the system is so efficient that the dimensionality collapses immediately. So this is a problem that we already encountered when we designed our electrophysiological experiments, and that's why it would be desirable to have a 3rd stimulus, so that here the subject cannot still decide the response. Okay, that's the only reason to introduce a 3rd stimulus. Okay, there are a lot of open issues that I only mentioned. We need to study more systematically the input fatigue. That's going to contaminate our estimate, but not too much, especially if the only thing you care about is to find the maximal dimensionality. There could be nonlinearities in the Ball signal. there could be a lot of fMRI noise, also given that you need to do p-square measures. I mean, you know, here you're trading precision with time, essentially. You want to be more precise, but you need to do more measures, p-square measures. So, you know, given a limited time of the experiment, then you need to consider this factor. Of course, you know, every time you do this repetition suppression, you have to be careful about expectation and attention effects. And it would be great to cross-validate all these results, and we still don't know exactly how to do it. Okay, so I need to thank a lot of people, and Mattia Rigotti is really the one who did most of the work, both for the first part and the second part of the seminar. Omri Barak, Marcus Benna, and Grace Lindsay, all at Columbia, they also contributed a lot to the theory, and we had some collaboration with Xiaojing Wang and Nathaniel Doe. The experiments, the electrophysiological experiments come from Melissa Warden and Herb Miller, who were at MIT when the experiment was performed. Now, Daniel Salzman, Randy Bruno, and Aniruddha Das are working on the electrophysiology, many different brain areas, and they also have intrinsic imaging, so then we can compare an estimate of the hemodynamic response to the electrophysiology. And recently, we started to interact with Calamit. For now, you know, we just discussed a lot about the protocols, but we don't have any results. Okay, let me finish with the second painting of Umberto Boccioni, States of Mind 2. If you want to see the third one, you have to go to the MoMA because I couldn't find it on the web. Thank you.
Very quick question. What do you mean when that the dimensionality drop means that there is not enough, so they cannot decide, so it's very short, the window in which they can decide which stimulus you can decode?
No, I mean, there we have 1 full second before the monkey can actually even know what kind of task it's going to perform.
Okay.
So it's not a problem of time. The problem is really that essentially the representation is low dimensional. I mean, I had many more slides on these. Let me see if I can find them. Okay, so here you see, you know, we use the same model to represent these things. And in this case, I have Q1 and Q2. Okay, so these are the representations of the 2 task-relevant variables. And now I'm going to have neurons that have nonlinear mixed selectivity. So they have both a linear component for Q1, linear component for Q2, and nonlinear mixed selectivity. And neurons that, just by chance, so because you learn this, they're highly selective to, let's say, 1Q. So then they don't have the nonlinear mixed selectivity component. Okay, and then you can have those that are selective to Q1. Okay, so these guys are those that are very important for the dimensionality, but they're also the most fragile to noise. So essentially, when these variables that are not under control start to vary in a much wider range, you lose this component. This becomes too noisy. Whereas these guys that are more specialized, they're much less sensitive to this kind of noise, and they will preserve some information about Q1 and Q2. So that's why you can still decode Q1 and Q2, because you can rely on the low-dimensional guys. These guys are not sufficient to perform the task, because you really need to mix things non-linearly in order to perform the task. However, you can decode individually Q1 and Q2. You cannot decode combinations of Q1 and Q2, but you can decode them individually. So that's our very simple cartoon for explaining what happens. We have no idea whether this is what's happening, but this model is compatible with the data. John?
Stefano, so I read the paper, the Nature paper, a couple of years ago. I don't remember the details now, but I'm a little confused about the key error trial analysis. So there you found the dimensionality effectively was lower. But what actually do you read out to say there will be an error? You're looking in that 800 millisecond interval after both stimuli have been presented, correct? And you were modeling the task where the animals made the two saccades. So it's not really a binary readout. So the assumption is if you find out that you're not encoding T1 and T2 properly, stimulus 1 and stimulus 2 properly, you predict an error, but you can't predict all errors, obviously, right?
Right. So we are considering this particular task, right, which is the recall task, and we're analyzing this interval here. So here, the monkey still doesn't even know what kind of task it's going to do. Actually, the monkey does because these are in blocks, but it still doesn't know what kind of stimuli will appear on the screen. Okay, so here the monkey cannot plan any motor response. However, all the information should be there. So this is the interval that we analyzed. Now, dimensionality, as you correctly stated, is a property of a set of points. It's not a single trial. So what we did was to collect all the points in the n-dimensional space that correspond to the error trials. We pulled them together and we estimated the dimensionality. Okay. And then we did the same for all the correct trials. So again, it's a property of a set of points. Now, with our definition of dimensionality, we can in principle define dimensionality in a single trial because it's related to the ability of the linear readout to decode, but we didn't do that analysis.
I hope you don't mind a very naive question about some of the modeling work that you started with. So you were discussing the advantages of having a mixture of nonlinear neurons at layer 1 because that makes essentially decoding of the outputs easier at layer 2. I guess my question is about layer 0 in a way. Does that need to have nonlinear coding at layer 1 create additional computational demands for the preceding layer that feeds into the representation of layer 1? So I guess the question is, you've got a complex problem, so have you just pushed it back into this preceding representation that feeds into your system?
Yeah, okay, so that's a very important issue. Here we focused essentially on one of the very last stages of the processing. Okay, so for instance, we completely ignored all the complications that you might have when you consider the entire visual system, where essentially what you need to do is to decrease the dimensionality. You know, you start from the retinal representation, and then you want to decrease the dimensionality because the dimensionality is too high if you want to do any generalization. Okay, so here we are focusing on a very specific problem, which is the following. Given that we already solved the problem of invariant representation. So now we don't care about the details of the visual object. We already have some kind of abstract representation of the object that we need to classify. Now we have a complex task that depends on a sequence of two objects. What kind of neural representations do you need? And in this case, all you need to do is to maximize the dimensionality. This is not always what you need to do. So for instance, you know, Jim DiCarlo actually is always very happy when he sees a reduction in the dimensionality, not an increase. Many people ask me, you know, do you get along with Jim? Because I actually do, because I mean, we're looking at different aspects of the dimensionality, and there is a very nice way of reconciling the two measures, but now I don't have time to show it to you, but given that we share the office, I can do it in one of the next days. But you're totally right. This is addressing only one specific problem, or one class of problems. If you want to address also the others, we can actually use the same techniques, but we need to change the type of analysis that I presented here.
Thanks.
Thank you.